Good afternoon, everyone, and welcome to our first session of Varsity Sci. As you may already know, Varsity Sci is the inaugural session of the symposium, which will hopefully be repeated in years to come, which is all about grassroots open science delivered from Oxford and Cambridge right to your doorstep. It's also all about bringing societies from Oxford and Cambridge closer together to collaborate. At this difficult time period, we thought that it was especially relevant and especially important that we get together and organize an event that reflects our commitment to science and to fostering a community of sharing research and innovation. So without much further ado, I'll just introduce how these events are going to work. So these events are going to be both streamed on YouTube, and you can also join in via Zoom by following the links to the forms, the registration forms in the event description on Facebook. What we're going to have is we're going to have a brief introduction for each speaker. We're going to have a talk of around 10 to 20 minutes, depending, of course, on the individual speaker. And what we will have after that particular talk is we're going to have a question and answer session that is going to bring in questions from both the Zoom conference call and from the YouTube live stream. So do feel free throughout the talks to ask questions in the chat box, and we'll pass them on to the host of that particular session. If you would, however, like to ask a question on the Zoom conference call, do make sure to message any of the members, the volunteers responsible. So for example, you could message me, Christoph, and I'm the president of Cambridge Biosoc, or my co-president Yising, and we'll be more than happy to either ask your question for you or to switch the video to you once the time for a question and answer session comes. So without further ado, I will now pass over to Yising, who will introduce this session and our first speaker.
Hi everyone, yes, I'm Yising, co-president of Cambridge Biological Society. So I'm the host for your session today, which has the theme Translational Medicine. We'll have 3 speakers today. I'll be doing the short introduction and I'll be also moderating the Q&A session. So first up, we have Rachel Foreman, who is a PhD candidate at the Institute of Metabolic Science at Cambridge. Her talk title is Peptidomics and the Hidden Messages in Obesity. Her PhD involves using mass spectrometry to measure peptides and gut hormones in various disease state patients, such as diabetes and obesity. She will be presenting some data from a recently published paper and findings from gut tissue analysis. Without further ado, let's welcome Rachel to deliver her talk. Thank you.
Hi, thank you, Yising. I'm Rachel, and I'll just bring up my screen. Okay. There we go. So yeah, as introduced, I'm at the Institute of Metabolic Science, which is down on the biomedical site at Addenbrooke's. And I specialize in mass spectrometry and sort of using this technique to develop and improve on measuring gut hormone peptides. So gut peptides are studied a lot in our department, and they're mostly secreted by the enteroendocrine cells directly into the bloodstream. So every time you have a meal, the signaling pathway between the lumen of the gut and the bloodstream, and then it secretes things such as insulin and glucagon to control your appetite and glucose regulation. So there are diseases such as diabetes and obesity where actually this signaling is interrupted, or the patients have lower levels or higher levels of certain peptides, which causes uncontrollable hunger in terms of obesity and poor control of glucose, such as in diabetes. So the clinical measurement of these peptides is actually really important to try and develop new drug targets. And clinical measurement is currently done by ELISA, which is an immunoassay method using antibodies which are targeting these peptides to then cause a signaling here. So this is just an example plate here of an ELISA. So you can see the wells with the darker blue will have a higher response of peptide than the paler one, and using a calibration standard you can quantify the amounts. However, these antibodies are actually being found to be slightly non-specific. So if you have peptides with similar structures, the antibodies can get confused, and that leads to a lot of misinterpretation of results. Whereas mass spectrometry uses slightly different measurement methods and is actually better at determining what the structures are and what peptides are present. So this is just a basic diagram of mass spectrometry, if anyone isn't aware. So following the pathway, the peptides are extracted from samples, and this is usually a plasma sample taken in the clinic. The extraction process then cleans up and removes any plasma proteins, and the small peptides can then be injected onto the liquid chromatography column, where the silica stationary phase has a affinity to the peptides of various peptides have various affinities, and as they are carried through in a mobile phase, this interaction causes them to be caught and then retained and slowly released into the mass spectrometer. So that's the separation part of the method. And then the mass spectrometer is the detection part. So as you can see, the samples are introduced and they are then ionized. I've just shown here that it's positive mode because the charged states of amino acids prefer to be positive mode. And then there's the separation in Q1. And Q2 causes collision energy to break down these peptides even further into smaller fragments. And then there's a second detection in Q3. And then as you can see, this is a chromatogram here for an example, which gives you a nice sharp response. And the peak area from this can then be taken and measured and used for the data analysis. So this is just an example I've chosen, which is a gastric inhibitory peptide. So this is secreted by cells in the gut lumen after glucose has been introduced to stimulate insulin secretion. So this is very important for diabetic patients. Now, as you can see by the amino acid structure here, it's in terms of peptides and proteins, it's relatively small. But the two initial amino acids, the red line there just shows the cleavage point, where an enzyme known as dipeptidyl peptidase 4 breaks off these two amino acids, and that essentially deactivates it. So this is a natural deactivation in the blood to sort of stop the stimulation of insulin once it's reached the important amount. However, the amino assay, because the amino assay targets the middle of the peptide structure, it can't actually tell the difference if these two amino acids have been removed. So it can't tell the difference between active and inactive GIP. But as you can see underneath, we've got the mass spectrum of these two peptides, so 1 to 42 and 3 to 42. There's a clear difference between the ionized forms. So you can see there's a shift in mass to charge ratio, and using these different values, I can tell the difference between the two peptide structures in plasma samples. So I keep talking about clinical studies, and as our department mainly use clinical samples from diabetic patients, these patients regularly have a glucose test. So they are fasted and then take either a glucose drink or a mixed meal test, which is a bit more of a smoothie. And then we can look at their glucose and hormone response over a certain period of time and determine if they're within normal range or if there's something interesting going on, potentially disease state. But I was interested in these samples because I was unsure if there's a difference between a glucose drink and a mixed meal test, so whether or not these changes in peptide content are actually also down to nutrient content, and it's the motility of glucose solution versus a thicker nutrient dense milkshake that could also impact this peptide response. So I had a number of samples handed to me by a clinician where they had seen a difference in response between the glucose and the mixed meal. So I performed a LCMS method looking for certain hormones which I thought would be interesting in this instance. And so this is just the data. It was 6 patients per data point, and the red is the samples from the glucose drink, and the blue is from the thicker, more nutrient-dense milkshake. And as you can see, the insulin response performs exactly as what we'd expect for both tests. So it both rises to the same amount, and there's the decrease as the body digests the food and the insulin is stopped for glucose production, glucose absorption, sorry. Motilin, conversely, has a different shape. Now this peptide, there is a very old immunoassay, sort of built in the 1980s, but since then nothing has been used to measure motilin. And motilin, as the name suggests, is a peptide that's secreted to help with the motility. So this helps with the muscular contractions of the lower intestine, so to pass the food along. And as you can see, when taking a glucose test, this is decreased immediately. So the motility is stopped in the gut to help with absorption of the nutrients, and then it slowly increases back to its homeostatic level. And what was interesting here is that you can see the mixed meal test and the glucose drink actually has different responses. So there's a slightly flatter curve for the nutritional milkshake, which suggests that the presence of these extra nutrients is actually having a slower effect, a slower regain to motility in the gut. And the last 2 graphs at the bottom are just the 2 GIP peptides, which I've said before are normally measured as a singular GIP, total GIP by immunoassay, but you can see that we have the 2 different peptides, and they actually go to different concentration levels. So this is because as GIP1 is being secreted and activating on the insulin, it's then immediately deactivated by the peptidase, and then that's when we get the increase in 3 to 42. So that's why there's a slight shift in 3 to 42 concentrations. And again, similar to the motilin, there's a different response between the glucose solution and the mixed meal test. So what's quite interesting here for me is that I can see that although the insulin response had the same peak due to the same glucose concentration, it's actually the other nutritional products in the milkshake, which would be fats and proteins, that have an effect on GIP, which will consequently have an effect on how the body absorbs glucose. So these are just the data points correlated against the immunoassay. So as you can see, as the mass spec can measure the 2 separate peptides, these correlate poorly against the immunoassay, whereas when you total them together and correlate that, that's a good correlation. And looking at the R-square values, it's actually higher for 3 to 42, which suggests that the immunoassay data from the clinic correlates more strongly with the deactivated forms of GIP, which is fine because we know that GIP slowly deactivates, and that's as it has a slower excretion rate for GIP 3 to 42. But this is just an example that there could be other peptides which are being measured incorrectly by immunoassay, and we're actually missing a lot of peptide activity there. So GIP is, I've talked about it, it's a very important peptide for insulin secretion and beta cell structure. And there's also GLP-1, which is secreted at the same time and is also important in insulin activity and cardiac output. So they're secreted from similar cells, so it's important to think that maybe they could be measured together, and the GLP-1 would give us more data from these OGTT and mixed meal samples. However, it's poorly measured by mass spec. So I figured, why not take what's good about the immunoassay and combine that with the mass spec detection. So as I've said, immunoassays use the antibodies to specifically target peptides, but can have cross-reactivity issues. So I developed a method to combine these methods where you apply the antibody bound to a bead to pull out the peptides from the plasma, and then by releasing the peptides, they can then be injected onto the mass spec and see if there's any difference between active and inactive forms of peptide. And this has previously been done in literature, and as you can see, they were able to measure both GIP and GLP-1 in the same plasma sample by binding to magnetic beads and using a pull-down method to pull out the two peptides for mass spec analysis. So this paper I used as my background to develop my own method to look for GLP-1 in patient plasma. So the immunoassay beads are readily available, and I also got a GLP-1 antibody, which was a monoclonal antibody specifically to target GLP-1. So this is currently being used in the immunoassay methods. So I knew it would be a strong affinity peptide, strong affinity antibody, sorry. And as you can see here on the sensorgram, you can see that the affinity to all of the different forms of GLP-1, so there's different structures here, is relatively strong. So after coupling the magnetic beads to the antibody, I then looked at the extraction. And this is just a mass spec chromatogram of proving that the antibody had bound to the bead. So I took some freshly bound antibody bead complexes and then tried to undo what I've just done, so remove the antibody from the beads just to make sure that the binding had been strong enough. And as you can see here, this is the amino acid sequence for the IgKC, so the immunoglobulin antibody was bound to the bead. So GLP-1 also is deactivated by the same peptidase as GIP, which is why they work so similarly in the body. And as you can see here, the different isoforms are easily recognized as two different products using the different charge states. So again, going forward, I would work with this charge state here, the 6+ and the 5+ for 7-36, as these give us the strongest response in the mass spec, so they're the most likely to give us the best chromatography. So this is the results from the first attempt at developing the method. So the antibody bead pull down method for 100 picogram per mil and 500 picogram per mil of each of the GLP-1 peptides in 250 microliters of plasma. So as you can see here, this chromatography could be improved. There's little to no background noise, so it's quite a strong response. You can't get confused between anything else in the sample. However, this could be improved as the homeostatic levels of GLP-1 are around 10 to 50 picograms per mil. So I knew it could be improved. And when I went and compared the bead method to other published methods for GLP-1, the antibody bead method was a lot lower than what also could be measured. So I think I was losing some GLP-1 somewhere in the sample or during the extraction process. So I then played around with sample volumes. So you'd think if you had more plasma, you'd get a higher response, but actually as the sample volume increased, it got to a maximum of 250 microliters, and then going higher than that, you'd actually have a plasma peptide cross interaction with the bead. So it's actually blocking the complex forming with GLP-1 in the samples. So I stopped at 250 microliters and went back to look at the mass spec side of the method. So after figuring out that I'd improved the extraction as much as I could, I thought if I could improve the mass spec detection sensitivity, that would give me nicer chromatography. So there are many things you can use for mass spec. So the mobile phase, which is in the LC method that flushes through. And supercharges are really useful reagents for when you only want to look for positively charged products, and they cause an increase in surface area for the positive charge to form on. So the droplets within the LC system are smaller, and then that increases your charging potential. And this paper here just shows that when you add this MMBA, metanitrobenzyl alcohol, as a reagent in the liquid chromatography, there's a shift in charge state. So you can see that it goes from measuring 543 to 6, 5, and 4. So I decided to add this to my LC method to try and see if I could shift the charge state and therefore improve my sensitivity. And it did work. So this is where my method is currently at. This was sort of some pre-lockdown data, so I haven't gone back to improve on this, but as you can see, I can now measure 25 picograms per mil of GLP-1 peptide in 250 microliter sample of plasma. So this is a normal volume of sample you'd get from a clinical lab, and I can now pull out 2 different GLP-1 forms and measure by mass spec, whereas the immunoassays are still combining these as total. So this is a vast improvement on what's currently being done, and going forward, hopefully I can apply it to some samples. I have a little bit more validation work to do. So I need to make sure that the antibodies and beads are stable and that they are selective. So I'm going to throw in some similar structured peptides to see if it just doesn't interfere there. And there's also a potential to get some JIT-bound beads and try and recreate what was done in the recent paper where they were able to measure both at the same time. Hopefully put this through my samples and be able to get some more information there, because I think there's more to learn from what's active and inactive, whereas the amino acids are getting a bit outdated now, and the mass spec can really gain some more insight into what's actually going on in the body once a glucose test is introduced. So yeah, just that's about it. That's 2 years work of method development, and it kind of doesn't show much when you only have a few chromatograms, but it takes a lot of effort, and I like to thank the rest of my lab for their help in coming up with ideas to improve the methods. So yeah, thank you. I'll take any questions.
Thank you, Rachel, for your interesting talk. So yes, just to remind you all, if you're on Zoom, feel free to post your questions, or you could send it to me privately. And if you're on YouTube, yeah, just post your questions on the live stream. So we have one question here. How practical is this method, in your opinion, in clinical practice, mostly in terms of cost?
So cost-wise, it's about equivalent, because the development of antibodies is the most expensive part of an immunoassay. And I think if you have a method that can measure multiple peptides in one go, rather than currently to measure all the study where I looked at all 4 peptides in one sample, that would have used 3 amino acids, and you only would have learned 3 of the important data points. So I think, yes, the mass spec is expensive to run, but I think you get a lot more information from a single sample as you would with current immunoassay methods.
Thank you. So I have one question. You did mention it was slightly challenging to detect the peptide using mass spectrometry. Could you just briefly summarize what the challenges are again?
Yes, so because of the concentrations for the peptides are at such low levels, and they are low levels when they're circulating in plasma. So if I was able to, and I'm working on some gut tissue samples where I can look at the peptides at site of secretion, where they're really high, whereas once you get the circulating plasma, which is more of a clinical sample, the levels are reduced. So it's just trying to get the sensitivity down low enough to quantify the peptide there from a clinical point of view.
See, yeah, thank you. So yeah, just waiting for more questions. And if you're on Zoom, feel free to unmute and ask questions in person as well. All right, there's one more question here. How common are the cleavage methods of activating and deactivating peptides in metabolism?
They're quite common, actually. They're the main way that your body sort of regulates activities. Obviously, if there was no peptidase cleavage going on, your body would keep producing insulin so much, and then you'd almost be like a diabetic patient, or the converse method where you have no insulin at all because it's not activating peptides, because the enzyme can also activate other hormones. So it's quite an important process in the body, and I think it's quite important to know at what point that's happening, because that could also be a point of the disease where your body is slow to respond to the hormones, and then that's where you can have increased hunger or decreased hunger. So.
Right, thank you. So if there are more questions, I'm pretty sure Rachel will be happy to answer them after any other talks. So I think right now we'll move on to the second talk. Thank you again, Rachel, for your wonderful talk.
Thank you. I'll stop my screen.
Okay. So next up, we have Aaron Kaur, who is an undergraduate at Cambridge. He'll be doing natural sciences. His talk will be on recurrent urinary tract infections, UTI. So he'll be presenting evidence from a large retrospective analysis of over 5,000 urine cultures collected and tested at a large tertiary care hospital in Singapore over a period of 5 years. So without further ado, let's now welcome Aaron to deliver his talk. Thank you.
Thank you. Let me share my screen. Okay, thanks everybody for coming to listen to my talk. So I'm Aaron, and I did this project over the past holidays at the Genome Institute of Singapore in collaboration with the National University of Singapore as well. And the main focus on this talk will be trying to understand more about UTI and urinary tract infections. So without further ado, I'll just get into the introduction. So why do we choose UTI as a disease to study? Well, UTI is actually one of the most common infectious diseases to humans. In fact, it has a very high morbidity and mortality rate. So in fact, almost 50% of women in their lifetime will get UTI sometime, will get UTI sometime in their lifetime. And there's also a pretty high mortality rate. It's 2.3% in the US, according to the CDC. And as well, my own country in Singapore, it is actually among the top 10 causes of death every year. So this is a slide I've taken from their website. As you can see, urinary tract infections actually rank 8th on the causes of death, which is quite significant. Generally, for UTI, E. coli is the most common uropathogen that is found in that contributes to UTI. And the main source of treatment for urinary tract infections is antibiotic therapy. However, as you all might know, there's a big global problem right now with rising antibiotic resistance across all areas of medicine, not just specifically for UTI. And the thing about the rising antibiotic resistance is it renders many frontline drugs used to treat UTI in the past ineffective. In addition, the thing about antibiotic resistance is that it varies from location to location. So that means that antibiotic resistance of UTI causing uropathogens, for example, here in the UK, might be very different from the antibiotic resistance rates for uropathogens in Singapore. Therefore, there's a need for local studies across the world so that so that local healthcare systems are better equipped to be able to understand and treat local UTI cases. In addition, patients suffering from UTI caused by these resistant uropathogens often are symptomatic for longer periods, and as well as have worse clinical outcomes, as shown by a few studies in literature. Therefore, it is quite important for us to deal with this big problem of antibiotic resistance. However, antibiotic resistance is not the only problem that that's not the only complexity to UTI infections. Recurrent UTI is also a very big issue when it comes to urinary tract infections, as up to 20 to 30% of women who suffer from UTI will end up getting recurrence, which, if you do the math, is about 10% of all women in their lifetimes might experience this once. Recurrent UTI is when patients have multiple incidences of UTI, and it negatively affects their quality of life. And this is likely due to two main causes. The first cause is that it could be due to ineffective treatment due to antibiotic resistance. So if the doctor prescribes antibiotic that the uropathogen is actually resistant to, then the UTI will be unable to be treated, and thus the patient will experience recurrent UTI. Another possibility for recurrent UTI, which has been brought up recently, is persistent intracellular infection, where the bacteria can actually attach to and then infect cells intracellularly in the bladder. So to illustrate this is a figure from research paper from 2007 on intracellular bacterial communities in humans. So these were initially discovered in mouse models, but similar populations were found in humans. And if you notice, what can happen is that bacteria can actually infect the cell intracellularly and hide inside the cell, which means that dormant populations of these bacteria might actually be existing in the bladder, but the urine culture, if any urine culture is done, it could be, there might be negative for any organism because all the uropathogens are hiding inside the cell itself. Thus, this leads us to a hypothesis which has been floated up in the community, that the slow growth and intracellular location of these bacteria actually result in low or no effective selection for antibiotic resistance, since when the bacteria are hiding inside the cells, they might not be exposed to the antibiotics, and as well as they might not be exposed to any of the immune system in our bodies. So this presents optimism in the case of rising antibiotic rates. Why do I say this? It is because not all recurrent UTI cases might actually be due to high antibiotic resistance in uropathogens. It could just be due to the fact that these uropathogens are hiding inside intracellular communities, and thus we think that we are unable to treat these recurrent cases because of their high antibiotic resistance. However, this hypothesis has been rather difficult to test. In mice, due to the long timescales of resistance mutations and the lifespan of mice, it is difficult to be able to observe any long-term evolution of antibiotic resistance. And in humans, due to the large number of variables which exists, so if you try to, it's quite difficult to be able to control for every single factor out there. And thus it has been rather difficult to test this hypothesis over the past few years. So what we try to do in this study is to try to lend some evidence that supports this hypothesis. So next, I'll be moving on to my data summary. So I'm just going to show you a big overview of our patient data. So initially, we obtained about 5,000 plus cultures from 3,000 plus female patients from a period of 5 years in a tertiary hospital in Singapore. And it's a retrospective study. So what we did was we analyzed records, past records from patients. And the first thing we did, we needed to be able to filter out our data So the first thing we did was that we excluded cultures where there was no organism observed at all, or there were duplicated cultures which could be perhaps due to administrative error, resulting in about but these two exclusions do not account for many cultures at all. So next, we try to take a look at the number of organisms in our data set, and we realized that we identified about 81 total different organisms. Here I show the organisms identified in more than 1% of cultures that we had. And if you notice, E. coli is the most common organism we isolated in our data set, which is consistent with many other reports around the world, where they tend to find that E. coli is the most common uropathogen. As such, we excluded cultures that are not E. coli, and we only focused on the E. coli cultures in our data set. This cut roughly about half the number of cultures from our data set. And lastly, because this data set is over such a long period, there were certain inconsistencies in the antibiotics where the cultures were tested against. So not all cultures were tested against the same antibiotics. As such, we limited the cultures analyzed to the cultures tested for antibiotics tested against more than 10% of our cultures. If this is a bit confusing, it should be clearer in the next few slides as I show you a few figures. So first, just to show you some demographics from our population, we have an age group from about 21 to 50. And as you can see, our age is actually pretty well distributed across all the age groups with a median age of 35 years old. And on panel B, you can see an ethnic distribution. And the good thing about this ethnic distribution is that it's rather similar to what we have in Singapore locally. So it provides a good representation of ethnic distribution in my own country where we are doing the study. Next, this is some variables from the cultures, variables associated with the cultures. So for example, we have patients who are inpatient, outpatient, and as for panel B, which shows where the urine was collected. So most of the samples were actually collected from the patient's midstream of the urine. And panel C shows the clinics where the cultures were isolated from. So as you can, I highlighted 3 specific clinics here, and I'll go into detail why later. And on panel D, you can see that the diagnosis which were associated with the cultures taken. So as you can see, there's a majority of our samples actually had no diagnosis given, which could likely just be due to physicians forgetting to input the diagnosis when they order the culture. And as such, we have a large gap in the diagnosis data. Next here, I present to you the resistance rates for our entire data set. So on panel A, you can actually see that the antibiotics that the cultures were tested against. So as you can see, not all bars add up to 100%. And as previously mentioned, that is why, not all cultures were tested against all antibiotics. As such, we have some limitations in our data set here. However, our resistance rates are generally similar to other studies, but it is difficult to make any strong observations about trends and resistance because of the lack of completeness of our antibiotic data set. And on panel B, you can actually see a histogram of the number of antibiotics tested. And the reason I show you this is to illustrate that actually for most cultures, it is actually tested against a majority of antibiotics. So it's not just tested against a few. And lastly, this graph shows the number of cultures each patient has. So this is on a log scale as well. This is on a log scale. And as you can see, a majority of our patients actually only have 1 culture. But what we are really interested in this data set is patients who have more than 2 cultures, which is why it's good that we have a large data set to be able to get a good number of people with more than 2 cultures during our time period. So next, I'll go through how some of the previous variables might actually have an impact on our resistance rates in our data set. So the first thing we wanted to check was that whether the UTI diagnosis was associated with resistance rates or not, considering that a large number of cultures actually did not have any diagnosis attached to them. So as you can see from this, all the antibiotics are actually not significant when we are comparing the resistance rates of patients with cultures coded with a UTI diagnosis and those without. So as you can see in this graph, I've only presented 8 commonly prescribed antibiotics for your convenience, but all 23 antibiotics are actually not significant in this area. And if you can see on the bottom, we performed statistical analysis was performed with a 2-tiered Fisher's exact test, and as a corrected test as well. So I also wanted to, it has been known in literature that patients from kidney transplant, urology, and oncology clinics might actually have a higher resistance rate. And that's what we wanted to check in our data set as well. So actually, which we found to be mostly true. So as you can see in our kidney transplant, urology, and oncology clinical locations, there's actually, for some of the antibiotics, there's actually a significant increase in the number of cultures who are resistant to the antibiotics. However, whether we included or excluded these data did not really have a major impact on the rest of our data, on our results. And as such, we included all cultures from these 3 clinics in the rest of our data set as well. So now, since I have covered some of the variables concerning our cultures and how they might affect our resistance rates, now I present to you our first major finding of this study, which is that there's no significant difference in antibiotic resistance rates in the first and last cultures of patients. So what we did is that we wanted to observe what was this resistance rates in the first culture of the patients compared to the last cultures of patients, to try to better understand whether this increase in resistance rates might show that might show whether intracellular location was helping or not helping bacteria to evolve resistance. So firstly, what we did was that we stratified our cultures by time. So we for all cultures less than a year and for cultures more than a year, we actually noticed that there's no significant difference between their first and last in resistance rates between their first and last cultures. And this is also true for the rest of the 23 antibiotics, which are not shown here. So there's no significance in the antibiotic resistance rates. And next, we also stratified the data by the number of cultures to ensure that number of cultures didn't really play a part. And we also found that the statement remained true, and there was no significant difference in antibiotic resistance rates in the first and last cultures of patients. We also checked for all the patient demographics mentioned earlier, such as age, ethnicity, and we found that the data was not significant as well. And as such, this result actually supports the initial hypothesis that I mentioned at the start of the presentation, where in the intracellular location, actually results in lower pressure to evolve antibiotic resistance for the bacteria. So what we found actually supports this hypothesis. However, there's some limitations as well. For example, the first and last culture of patients within our data set is just within our data set. So we're not really sure whether patients might have earlier incidences of UTI or later incidences of UTI. But as our data set is rather large, spanning over 5 years, we feel that it is a good representation nonetheless. Another limitation is, of course, the inconsistent testing of samples of antibiotics, as I mentioned earlier, and as well as the lack of data on antibiotic therapy in this data set, which might have been helpful to understand how specific patients were actually responding to the antibiotics that they were given. So now, since we kind of understood the resistance doesn't seem to change within a patient during our study, so we wanted to also find out what effect does antibiotic resistance actually have for UTI. And what we found was that actually the antibiotic resistance in a patient's first culture was associated with a patient having multiple cultures in our data set. So in this figure, we compared whether patients initially had a sensitive or resistant uropathogen culture. And then what we did was we checked the percentage of patients who went on to have multiple cultures or single cultures. And as you can see from the data, for a majority of the antibiotics, and for the rest of our data, it's 17 of 23 of the total antibiotics, there's actually a significant association, a significant increase in patients who have multiple cultures, who are initially resistant, who have initially resistant bacterial cultures. As you can see, for example, for like amoxicillin, we don't have actually data of resistant cultures at all, which leaves us unable to be able to conclude anything. And as well, for many of our not significant, or data which has not significant, for example, like nitrofurantoin, the issue we kind of have is that we do not have a large number of cultures which are resistant to it. So we are unable to, so we think that could be why we are unable to really observe any significance in our data set. So what this result shows us is that it lends support to the hypothesis that there's association of higher virulence with resistant bacteria. So then this could be due to resistant bacteria causing complications with treatment, and as well as association of genes responsible for increased virulence with resistant genes. And we also have rather similar limitations to this result. And especially the issue on uneven sample sizes, which really kind of limits us in trying to find out if some of the antibiotics were really significant or not. So in summary, what we have actually covered in this study was, firstly, we presented a really large data set from a hospital in Singapore. And as mentioned, it is very useful to update local trends in antibiotic resistance and to be able to guide local empirical antibiotic therapy in Singapore. But globally, this also helps the scientific community as we show evidence to support the hypotheses about potential drivers and consequences of antibiotic resistance. And this also helps present a rare potential for optimism in the big crisis that's facing us, that is antibiotic resistance. So I would like to thank you for attending my talk, and I would like to thank my lab in Singapore, who really I learned a lot from them, and they give me a lot of support throughout this process, and especially my supervisor, Dr. Swain Chen, and as well as the many collaborators we had in the National University of Singapore. And lastly, I would like to thank the Varsity Sci team as well for giving an undergraduate like me an opportunity to present science. Thank you so much. Please feel free to ask any questions, or if you have any further questions, you can also contact me via email or LinkedIn. Thank you.
Thank you again, Aaron, for the talk. We appreciate your submission as well. And yeah, I hope everyone enjoyed your, thank you for that talk. So I have one question, my own question. So I'm just a bit unclear as to why, so is the number of cultures for each patient directly related to just the sample, the number of samples collected from each patient?
Yeah, so each sample is, so each sample collected from patient becomes a culture. So I've used the words interchangeably.
Okay, I see.
Yeah, they mean the same thing. Sorry for the confusion.
So for example, for if one patient is has only participated in the study for, let's say, just less than 1 year, then maybe they will have less samples, so that's why they have maybe just one culture. Is that correct?
That is unlikely to happen, because what we actually did from our data set was that, because it's a retrospective study, what we did is that we the study obtained permission to go into the medical records from the hospital over the whole period of 5 years. So we put all the data from hospital records over the 5 years. And so it is unlikely that a patient might say, like, stop participating in the study. But what could likely happen instead is that a patient could have perhaps gone to a different hospital during the study, and we might not have picked up a culture then. But we also think that is unlikely, because in the Singapore healthcare system, generally people tend to go back to the same hospital for the same condition.
I see. Right, thank you so much.
Thank you.
And there's one question. Why do you think there were no amoxicillin-resistant cultures?
Yeah, that's a great question. So the thing about our data set is, because it's over such a long period of time, many of the antibiotic like the antibiotics that the cultures were tested against were highly dependent on what the physician ordered. So for example, the doctor might have specified certain things. And I think in our data set, amoxicillin was actually, amoxicillin, as you can see in this slide, is actually quite a small percentage. So like less than 20% of the cultures were actually tested against amoxicillin. And that could be why we actually don't observe any resistant cultures at all, because it's just tested against such a low number that we didn't manage to catch any resistant cultures.
Thank you. Another question is that, so from your data, samples from certain clinics, for example, from kidney transplant, urology, and oncology, show a higher antibiotic resistance rate. Would it be possible that it's because patients from those clinics receive more antibiotic treatments compared to other patients from other clinics?
That could be one of the cases, but we didn't explore this option too much on why exactly these patients had higher resistance rates, as it's not really the focus of the paper, or the focus of the study rather. But it has actually been well documented in literature out there that these patients actually do have a higher resistance rate. And we think that it could be due to a variety of reasons, including maybe these patients could be immunocompromised from their treatment, or many other variables such as the one you suggested.
All right, thank you. So one more question. Are there plans for further research, including differentiating between stages of antibiotic treatment?
That could be one area where we might look into further next time. But for now, with the constraints of our data set, this was what we managed to do. Of course, perhaps in the future, we can and we should definitely look into other things like what you suggested. So thanks for the suggestion.
All right. So we'll wrap up for your talk now. Thank you again.
Thank you.
Our next speaker is Rebecca Jones, a PhD student at the MRC Epidemiology Unit. She'll be sharing the main findings of a recent systematic review, which aimed to find out how attending a weight loss program affects the mental health of adults with obesity. Her team looked to understand how different aspects of mental health were impacted, including anxiety, binge eating, body image concerns, depression, distress, emotional eating, negative emotions, quality of life, satisfaction with life, self-efficacy, self-esteem, and stress. By better understanding how mental health is impacted, future programs can be developed to more effectively support both physical and mental health at the same time. Without further ado, let's welcome Rebecca to deliver her talk.
Fabulous, thank you. Let me just share my slides. There we go. Yeah, so as you just said, so I'm Rebecca Jones and I'm a final year PhD student at the MRC Epidemiology Unit, Cambridge, and I'm going to share the recent findings from a systematic review, which forms part of my PhD, which assesses the impact of weight management interventions on mental health. Oh, I'm on the wrong thing there. There we are. So you can see that properly now. So, as I said, this review forms part of my PhD, which is the overarching aim to assess the impact of behavioural weight management interventions on mental health and to understand how to better support adults with obesity with their mental health. My presentation today will go through the key why, what, how, and so what questions. And this will be more of an overview talk, but the full review has been accepted at Obesity Reviews, so we can see more detail and more in depth there. So, behavioural weight management interventions are the most common form of obesity treatments in the UK and are recommended across the world by national guidelines. These interventions are usually multi-component and they target changes in diet and physical activity through behaviour change support with the overarching aim of supporting adults with obesity to reduce excess weight, minimise health risks and improve health outcomes. Weight management services prioritise improving people's health by reducing risk factors without a sole focus on weight, but this is rarely the case when services are evaluated for effectiveness or when they're assessed for a commissioning value. In addition, the research landscape provides a good evidence for the effect of these interventions on physical health. However, there's very limited and inconsistent evidence investigating mental health related outcomes and the unintended effects of these interventions. Which raises the question, can we truly recommend an intervention to be implemented if we're unsure of how it's going to affect the mental health of those that we're encouraging to attend? So previous systematic reviews have assessed the impact of weight management interventions on mental health and have reported inconsistent findings, as you can see on the slide. The green arrows represent reviews which had beneficial findings for mental health, and the red arrows represent reviews that either found no difference or a detrimental impact on mental health. So you can see that it's really contrasting findings, and it makes it really difficult to get a clear understanding of how interventions are affecting mental health as a whole. And this is made more difficult by the limitations of previous reviews too. So previous reviews have applied very restrictive criteria. So some reviews included only successful trials, have only included diet only, exercise only, CBT only, have limited participants to what they have referred to as uncomplicated patients only, and have studied a very limited number of outcomes as well. So in the process of developing the review protocol, it was really important to us to address these limitations as best as possible, because we aimed at providing a more comprehensive overview of the impact of interventions on mental health. So we'd identified that there was a lack of up-to-date comprehensive review, investigated the impact of weight management interventions on a broad range of mental health outcomes in a representative sample of adults with overweight and obesity. So we overall aim to evaluate the impact of these interventions on mental health and determine if these effects are different depending on intervention or participant characteristics. So today I'm going to focus on sharing the main review findings, being the impact of interventions on mental health, which we assessed at 2 time points, at the end of the intervention and then 12 months after starting the intervention. So the review process began with defining the eligibility criteria. So as you can see here, we kept a focus on study quality, representative populations, and replicating intervention characteristics to what's commonly delivered within Tier 2 weight management services. We applied a few exclusion criteria to ensure that the review is deliverable and achievable. We excluded populations with specifically one physical or mental comorbidity or pregnant women. We excluded eating disorder, surgical and pharmacological interventions to make sure we're focused on those that are most similar to Tier 2 interventions. And we also excluded theses and non-English language papers to constrain the search and ensure that the review was achievable. We completed the search in 7 databases on the 7th of May, and after removing duplicates, we title and abstract screened just under 32,000 papers.
We used confidence systematic review software for screening, and it resulted in 43 papers meeting inclusion for the review. Data was extracted from these 43 papers using a modified Cochrane data extraction form and the Cochrane risk of bias tool. So the data that we'd extracted, we then meta-analysed by outcome, and we applied meta-regression where possible to identify where potential sources of heterogeneity were in the results. I also completed sensitivity analysis to identify if the risk of bias ratings influenced the overall findings, and conducted stratified analyses to explore if the effect was different for key characteristics, for example, face-to-face interventions versus online-only interventions. But today I'm going to focus on the key meta-analysis findings, and I'd recommend downloading the full paper to see the more in-depth and detailed results. So, the more interesting bit of the findings. So of the 43 included studies, most studies were conducted in the USA, UK or Australia, and most of the participants were between 31 and 50 years of age, most were female and had a BMI between 30 to 39.9. The majority of interventions provided were education only, which may have been in the form of behavioural education sessions, online information, or behavioural counselling. Most interventions lasted between 2 to 6 months and were delivered face-to-face and on a one-to-one basis. We created 25 different meta-analyses for the different outcomes at the end of the intervention, and almost as many for the 12 months from baseline time point 2. And evidently this is far too many numbers and far too many graphs to be able to show in one overview presentation. So today I'm just going to talk you through the main key findings of the review instead, and focus on how these may be able to influence future practice and research. So at the end of a weight loss intervention, we found small improvements in depression, mental health quality of life, and self-efficacy. We found little to no difference to anxiety, overall quality of life, self-esteem, or stress. And there was too little evidence to make conclusions about the impact on body image concerns, emotional eating, negative emotions, distress, or satisfaction with life. 12 months after starting the intervention, we found small improvements in depression, mental health related quality of life, and self-efficacy, and little to no difference to overall quality of life. And you can see that we had too little evidence to assess the impact on many outcomes at 12 months from baseline. And these were anxiety, body image concerns, emotional eating, distress, negative emotions, satisfaction with life, self-esteem, or stress. So overall, we aim to assess the impact of behavioural weight management interventions on mental health at the end of the intervention and 12 months after beginning. And we found attending a weight loss intervention may slightly improve some aspects of mental health. And importantly, we didn't find any evidence to suggest that interventions negatively impact mental health. It's important to note, though, that meta-analyses assess the average effect on interventions, and it's likely that this isn't the case for all participants. We really need more high quality studies, especially where they're measuring multiple different types of mental health outcomes over longer durations in time, to have a clearer understanding of the impact on mental health. So while the evidence suggests that on average, mental health outcomes improve following a behavioural intervention, it's likely this isn't the case for all participants, and it's vital that we're able to identify those who are at risk of detrimental impacts, so that we can apply sufficient intervention in a timely manner. We also need studies that are sufficiently powered to investigate the impact on mental health in different subgroups of participants. But it all comes down to the fact that we really need many more trials to measure a variety of outcomes over longer durations and prioritise transparently reporting the findings. So this work could not have been completed without the help of my brilliant co-authors, which are shown on the screen, or without the support from my funders at the MRC. Thank you all for listening. I'm happy to answer any questions, and my email's on the slide if you have any further questions or comments for afterwards. Thank you.
Thank you, Rebecca, for your talk. So I'll start with one question. Is BMI still used as a common measure for something like patient fitness? Because I think there is, or like there are some arguments about how good this measure is, because you can't really tell the proportion of, let's say, muscle to fat by just looking at the mass of a person.
Is it BMI that you're referring to?
Yeah.
Yeah, so that's not what I kind of focus on. I focus on more mental health related measures, but the physical measures, there are definitely pros and cons, and there's different populations that they are applicable, and there's populations that they aren't applicable to. BMI can be useful for the majority of the population. There are subsections of the population that it's not really applicable to, and it certainly shouldn't be used on its own. It's a one factor, just in the way that when I talk about mental health, if we just focus on depression, it doesn't really give us an overall picture, because there's stress, there's mood, there's anxiety, there's body image. Just looking at BMI isn't enough. You need BMI along with other, like a package of factors to assess. So it can be useful, but it has to be useful with a full light in it, knowing the pros and cons.
All right. Yeah, could you give an example of patients or people who whose mass or whose fitness isn't very measurable by BMI?
So elite athletes is a primary one. Rugby players are often not great to be using BMI on. Those with a huge muscle mass, a lot more than the average population, being elite athletes are the main one that I would say. But also that definitely isn't my specialism area. I focus on mental health, so I wouldn't claim to be the specialist in that kind of thing.
Right, yeah. Okay, thank you. One question here. What is your take on the government's recently announced strategy to combat obesity?
It's very political, isn't it? I think there's pros and cons. There's ways that they need to improve. I think it's very positive that there are things being done. I think that it was timely that we needed to get more talk into the environment and into the community, and to it being a more political conversation. It's good to see it being a political conversation and to start having a drive towards treatment and care. But there's certainly ways that it needs to improve. As always, with every kind of a political agenda, there's ways that they can be improved.
Thank you. Another question from YouTube. Does the gender balance in the review reflect the gender balance of obesity in the general population?
So I wouldn't say it reflects the general population. It is very representative of interventions, though. There's engagement issues and attrition issues within weight management interventions in the community and in like the real world. For males, there's a very, very low rate of males within weight management interventions, and that's a whole, I know a lot of people that are doing work on that in itself, of male-only health interventions. So I would say that what we found in the review is representative of interventions populations, but not representative of the whole population. So what we were aiming to look at is representative, but there's certainly issues that need to be addressed with improving attrition and making a balance of interventions versus population.
Thank you. There's one question. How could we highlight potential negative aspects in such reviews?
Potential intended impact.
Yeah, potential negative impact on mental health.
I think it really does come down to measuring it more often, and it being understood that it's just as important as physical health, especially so within weight management interventions. It's very common to have a whole battery of physical health tests, and it's not usually that common that people measure mental health, despite there being a very known link between obesity and mental health. yeah, I think it just needs to be more of a conversation, and that it needs to be measured more often and transparently reported, because it is quite often reported that they have measured it, but then the actual data and the outcomes isn't reported in the results papers, which is a multitude of factors. It comes down to journals and funders, and it's a whole complicated multi-level problem. But yeah, I'm happy to see that it is changing a little bit. I think it's becoming more of a conversation, and it is becoming more evident that people are aware that focusing on physical health can have mental health consequences.
All right, I think if there are no more questions, we'll wrap up the session today. So thank you very much, Rebecca, for your talk. And I'll pass the floor to Christoph to do the wrap up.
Thank you.
So if I can once again thank all of our speakers today who have covered, even this one single session that's opening our symposium, who have covered a range of topics from the effects on mental health of weight management treatment through antibiotic resistance, through to the use of mass spectrometry combined with ELISA-style tests. So we've had a plethora of topics covered in this one single short session, and I hope that you'll be able to join me for further sessions of Varsity Sci. So our next session will be at 2:30 p.m. Of course, this is in British summertime, and this session will be on cancer. This session will be followed at 4:30 p.m. by our keynote, Professor Nasmith. And after that, we'll conclude today with a session on virology at 7:30 p.m. So I hope you'll join us on the live stream shortly. And in the meantime, thank you once again to our wonderful speakers today. And thank you for watching, and thank you for listening both on YouTube and through Zoom. Okay, thank you very much, everyone.